Hello students, this is Ms. D.M. Ivekar working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. In the last lecture, we have just discussed about how the pyruvate which is formed in the glycolysis is converted into the acetyl-CoA. And now in this lecture, we will study in detail about the Krebs cycle or TCA cycle. That is what happens to that acetyl-CoA when it will enter into the mitochondrial matrix. Now, why it is called as a Krebs cycle? Because scientist Hans Kreb has discovered this cycle in the mitochondrial matrix. Why it is called as a TCA cycle? Because if we will study this cycle, the first product of this cycle is having the 3 carboxyl group, that is this one. So it is also called as a tricarboxylic acid cycle. Now, what will happen to the acetyl CoA? Or what is the main application of this Krebs cycle? As this Krebs cycle will take place in the mitochondrial matrix, here there is a generation of this NADH and FADH. That is the energy sources. The one fact is it will generate the energy in the form of oxidative phosphorylation. Again, the second is it acetyl CoA which will enter in this cycle. It is also used for the synthesis of the fatty acid. The next one is this whatever the intermediates are there in the Krebs cycle that are also form the skeletal framework for certain amino acid. For example, this alpha ketoglutarate. From this, the glutamic acid can be synthesized whenever it is required. So this is a very important pathway or very important cycle which takes place in the mitochondrial matrix in case of aerobes, and in case of anaerobic organisms it will take place in the cytosol or that cytoplasmic matrix in the cell. Now, the simple funda, this trick is not designed by me, but when I was studying this cycle, I got this trick and I still remember that trick. So, we have to also remember this trick so that we can draw this cycle in very easy manner. Now, first, this acetyl-CoA when enter into the mitochondrial matrix with the help of a citrate synthase enzyme, it will converted or it will attach or it will react with this oxaloacetate. Oxaloacetate is a 4 carbon compound, and this acetyl CoA which is a 2 carbon compound, it will react with the oxaloacetate in the form of an enzyme with the in the presence of an enzyme citrate synthase, and this citrate synthase enzyme will form citrate. Now this citrate is having a 6 carbon unit. This citrate is acted upon by the aconitase enzyme, and it will convert this into the cis-aconitate form. This cis-aconitate form will get converted into the isocitrate form, and this isocitrate is also in the form of a 6 carbon compound. So this is the cis-aconitate which is the intermediate step. Now, this isocitrate in the presence of isocitrate dehydrogenase enzyme, always remember whenever there is a dehydrogenase term in the enzyme, surely there will be the formation of a NADH or FADH, surely. So here, this isocitrate dehydrogenase enzyme will act on this isocitrate and it will remove one carbon dioxide. It will also form one NADH here. So, isocitrate will form the alpha ketoglutarate here. This alpha ketoglutarate will be acted upon by the alpha ketoglutarate dehydrogenase enzyme. Here, the reduction of NAD takes place, and this alpha ketoglutarate will be converted into the succinyl CoA. Here also one carbon dioxide molecule is removed. So, this 5 carbon compound will be converted into the 4 carbon compound. Obviously, isocitrate was in the form of a 6 carbon compound. Alpha ketoglutarate is in the form of a 5 carbon compound, and then it is converted into the succinyl CoA which is a 4 carbon compound. Now this succinyl CoA in the presence of a succinyl CoA synthetase enzyme will be converted into the succinate, and here there is a formation of or there is a substrate level phosphorylation. Here we will get the GTP molecule. Now this succinate will be converted into the fumarate in the presence of succinate dehydrogenase enzyme, and this dehydrogenase enzyme will reduce this FAD molecule into the FADH2, right? So the fumarate, which is having a 4 carbon unit. Now the fumarase enzyme will convert this fumarate into the malate one. This is also the 4 carbon compound. And in the presence of a malate dehydrogenase enzyme, the malate is converted into the oxaloacetate. Now, if we'll if we will identify the number of NADH which are formed in this cycle, then one NADH is formed at level of isocitrate dehydrogenase enzyme. Another NADH is formed at alpha ketoglutarate dehydrogenase. The third one is at malate dehydrogenase. That is, a three NADH are formed in this cycle. Then one FADH2 is formed at the level of succinate dehydrogenase, and one GTP is formed at the substrate level phosphorylation at succinyl-CoA synthetase. Now, how you will remember each and every step so that there will be no any mistake during drawing this cycle. So you have to just remember this trick, that is, an Australian cricket club is OK and successively successful future matches OA. And Australian A for acetyl-CoA, cricket C for citrate, this C is for cis-aconitate, this E is for isocitrate, this OK and means alpha, OK K for ketoglutarate, so alpha ketoglutarate, S for succinyl-CoA, this second S for succinate, then F for fumarate, M for malate, and OA, that is oxaloacetate. So Australian Cricket Club is OK and successively successful future matches OA. And from that, just we will focus on at which step the NADH is formed, at which step FADH2 is formed, at which step there is a substrate level phosphorylation, and at which step the carbon dioxide is removed. Then this is the very nice cycle to remember. So now, what is the meaning of a oxidative phosphorylation and substrate level phosphorylation? It is very simple. At the during the metabolic process, the during the reactions, wherever there is a direct formation of the triphosphate is there, that is ATP or the GTP, then that type of a phosphorylation is the substrate level phosphorylation. And from that, we can directly or cell can directly utilize it as an energy purpose. But what about the step at which the energy is formed, but it is in the form of a reduced molecule, that is NADH or FADH2. In that case, that NADH or FADH2 which are formed, it will get transported to the electron transport chain into the mitochondrial region, and then from that we will get the energy. So there is a no any direct use of that molecule as an energy purpose like the ATP or GTP. First, in order to get the energy, that NADH or FADH2 has to be transported to the electron transport chain. So that is the oxidative phosphorylation. Now, during Krebs cycle, number of intermediates are used for the different metabolic pathways. As I have already told you that the alpha ketoglutarate like intermediates can be used for the synthesis of the glutamic acid. So, number of molecules are used. So, these reactions are also called as the anapluretic reactions. Now, we will study about the energetics of a TCA cycle. That is, how much energy we can generate from the when the one pyruvate is entered into the Krebs cycle. First, the pyruvate was converted into the acetyl-CoA at the mitochondrial membrane level. So there is a this is the oxidative phosphorylation because there was a generation of a NADH, right? So we will get the 3 ATP when it was transported to the electron transport chain. The second NADH was at isocitrate to oxalosuccinate level. There also we have get one NADH. Then the third one is alpha ketoglutarate to succinyl CoA. There also we have the 3 ATP, that is one NADH. Then succinyl CoA to succinate conversion was generated one GTP, that is called as a substrate level phosphorylation. Then succinate to fumarate conversion was there where there was a formation of a FADH2. From that we will generate 2 ATP, right? and then from malate to OAA, oxaloacetic acid, the cell will generate one NADH. So at overall, the 14 ATP from the oxidative phosphorylation step and the 1 ATP from the substrate level phosphorylation, 14 plus 1, that is a 15 ATP per pyruvate. There is a synthesis of the 15 ATP per pyruvate molecule. But from the glycolysis there are 2 pyruvate, so 15 into 2 we will get the 30 ATP. So 8 ATP from the glycolysis and 30 ATP from the TCA cycle. 30 plus 8 will gives 38 ATP that we were usually writing whenever we were writing the equations for the photosynthesis or the respiration mechanism. So this is the main cycle of generation of the energy inside the cell, which will oxidize glucose completely, and number of ATPs are generated at the some of the ATPs are generated at the substrate level phosphorylation, and most of the ATPs are generated by the oxidative phosphorylation. So in this small video we have discussed about the Krebs cycle. We have discussed also about the trick to remember the cycle, and we have discussed about the number of ATPs which are generated in the TCA cycle. I hope that you have understand this lecture. Thank you. Thank you for your patience listening. Thank you so much.